Let's review everything we learned in cellular respiration. So we start with glucose. Cells taking glucose, and it's going to go through glycolysis. It will make some pyruvates, and when oxygen is available, the pyruvate will go into the matrix of a mitochondria. And while going in, it's going to lose carbon dioxide and all that. Acetyl-CoA, it will turn into acetyl-CoA, and then it's going to meet up with oxalic acetate and go through this citric acid cycle or Krebs cycle. And then all the NADHs and FADH2, they're going to go drop their electron over here. The electron will go through ETC, and that's how we make ATP, okay, with chemiosmosis. Now, when there's no oxygen available, the pyruvate cannot enter mitochondria, and it's gonna, depending on what organism you are, it's gonna turn and go to what is it, alcohol fermentation or lactic acid fermentation. Okay, so that's it. Next, let's talk about the 4 steps here. Let's review this. We already talked about it in detail. So you have glycolysis, making 2 pyruvates. Doing so, net-wise, we're gonna get 2 ATP. Remember, this is substrate level phosphorylation here. Okay, and we're gonna make NADHs, and that's gonna go into oxidative phosphorylation. Okay, and intermediate step, pyruvate oxidation. No ATPs made here, but you make 2 NADH. Why 2? Because we're accounting for 2 pyruvates going in. Okay, so remember that. And then you got 2 acetyl-CoAs, meaning 2 cycles of citric acid will happen per 1 molecule of glucose. Okay, so everything gets multiplied by 2 here inside mitochondria. Okay, because we got 2 pyruvates. Now, so 6 NADHs from citric acid cycle, 2 FADH2 from citric acid cycle, and substrate level phosphorylation, you make 2 ATP. Okay, and then all these energy-rich electron carriers will participate in ETC, electron transport chain, and chemiosmosis, and will make about 26 to 28 ATPs. So total is about this many ATP molecules for 1 glucose molecule.